Uh, Sorry, it was just telling me that I was being recorded.
Oh,
So I'm Chris Curran. I'm at Northern Kentucky University. I made it to full professor here, and I'm now director of our neuroscience program. And early acknowledgements to KBRIN/Kentucky INBRE, who provided the funds to get my first R15 on polychlorinated biphenyls. Bridge funding to my current grant looking at traffic-related air pollution and benzoate-pyrene. And has supported a postdoc and a research technician. So I left that off the slides, but definitely have to acknowledge the funding. So technology willing, it will let me change slides. Good. So acknowledging the students, I added these up and I was like, oh, I must have been here a while. So we're at 95 of Team Norse, undergraduates and one high school student. And we're a very productive lab, as you can see. I've had 4 students who actually, 3 of them during their undergraduate career get married and have babies and 1 since. So we're into developmental neurotoxicity and healthy babies. My thought for the day is shared from my F1. So Melinda Curran MacDougall has been a collaborator on some papers. She's a SAS programmer and knows a whole lot more about statistics and analysis than I do. And she sent me this when I was struggling. And it was like, oh, okay, the reason I have all questions and no data is I haven't gotten to part 2. So our F2s are down here. So Brandon and Allie already are showing very, very strong interest in science, particularly Allie. Brandon seems to be more the analytical side. So in the lab, why are we worried? Because we want healthy babies, healthy moms. I am a member of the Society of Toxicology and the Developmental Neurotoxicology Society and the Society for Birth Defects Research and Prevention. So I think you can tell that this is definitely a personal passion. And these are some of my colleagues who have graciously shared their photos. So another question, why are we focused on genetics? Because we know there are just tiny differences that can mean a lot. The example I always give is we know someone who can drink a pot of coffee and go right to bed. The other person has a cup of coffee after lunch and they're up all night. It all has to do with genetic differences in our metabolic pathways. Why do we focus on the environment? Well, as some of you who signed on early heard, I'm from Cleveland. So I grew up under brown skies and the fish, so I've been interested in the environment from way back. But these are exposures that we have very little control over. And that's our main interest. So widespread and persistent pollutants. Benzoate-pyrene is found in vehicle exhaust, cigarette smoke, grilled foods, and we've certainly seen a lot with the wildfires. So there's tremendous amounts of exposure. And so it's a widespread risk. Why do we use mice in the lab? Well, they share a lot of our genetics. The mice we have have 3 genes that are orthologs to the human version, so it's a very good match. And in terms of undergraduate research, these are examples. Mice do relatively simple things when we test their learning and memory, their motor function, their anxiety and depressive-like behavior. And all of these lend themselves really nicely to undergraduate research, which is why you saw so many represented. Our pathways of interest, this is where probably the most technical slide I have. The aryl hydrocarbon receptor, the AHR, is a transcription factor that will bind to those pollutants. So both polychlorinated biphenyls, dioxins, benzoate-pyrene. Once it binds that ligand, it moves into the nucleus, has a binding partner, and then it will upregulate and it can downregulate hundreds of genes. The canonical pathway upregulates 2 metabolic enzymes, CYP1A1 and CYP1A2. So cytochrome P450 is their formal name. I will refer to them as the CYPs 1A1 and 1A2, primarily found in the liver, but also expressed widely in the brain and tissues. A key thing about the aryl hydrocarbon receptor is there are genetic variations. So some readily bind to these lipophilic ligands and others have weaker binding. So the amount of gene expression and gene up and down regulation can vary tremendously. These are the differences in our mouse model. So we have 4 different lines that we have been testing. The focus today will be on these first 3. So CYP1A2 is present or absent. We focused on that first because of earlier research with dioxins and PCBs indicated that if the mother lacked this gene, more of the pollutant then transferred in breast milk and during pregnancy. So we went with what had been identified as the most susceptible, and we're currently working with the 1A1 knockouts. The AHRB and the AHRD are the ones that either have very high affinity and will readily upregulate, and then the poor affinity takes a much, much higher dose before you would get upregulation. The other way I can show you that genotype matters is by looking at what happens to benzoate-pyrene. So it's not the parent compound that's the problem, it's the reactive metabolites. And those are highlighted here with the red circles. These are arene oxides, epoxides, and they can bind to DNA. So BAP was first identified as a carcinogen, and we're studying it in terms of its teratogenic effects. There are other pathways, though, that can lead to detoxification, and those pathways are identified in green. Because there are multiple pathways, we have to look at all possible genotypes to identify the most susceptible versus the most resistant. The 2 specific questions that we're focused on is if BAP exposure during pregnancy and early life will permanently change the brain. And then we want to identify those at highest risk, because when you have widespread pollutants, it's very difficult to protect everyone. So if we can identify the subpopulations when we get to the intervention step, that's going to be important. Experimental design is fairly simple. So we have the students we treat with corn oil soaked cereal to avoid any stress on the dam. So it's a treat they love. We train them to eat peanut butter Captain Crunch, and they'll just gobble up their daily dose. And we are going to measure those BAP and metabolite levels at the highest exposure. So they've gone through pregnancy, they're dosed all the way to weaning. Then we do the behavioral phenotype, which I'll talk the most about at early adulthood, which is postnatal day 60. And then when they're done at around postnatal day 120, we'll look at their neurotransmitter levels. The key behavioral findings, hot off the press. I just got the email today that the printed versions went out. This was online in early December. So again, you can see it was quite a group, including Greg Hawk, who works with Arnie Stromberg down at UK. So he did the statistical analysis on this. Just some key findings. And happy to share the paper with anyone that wants to see the details. We looked at open field locomotor activity. This can show hyper or hypoactivity, but normally mice are going to hang out on the periphery and not be in the center. This is standard behavior, and we definitely found that the poor affinity AHRD knockouts showed more of this anxiety-like behavior. So they stuck to the periphery compared to the other groups. Less time in the open. So this is really sort of our hallmark, looking for that genotype that is most affected. So that was significant. When we looked at motor function on the rotorod, which spins up to 20 revolutions per minute, and we test them over 5 days, how long does it take before they fall off? So those that have a shorter latency to fall are the ones that are showing the impairment. And in this case, it turned out it was a sex by treatment interaction, and the BAP treated females were most impaired. We also looked at learning and memory in 2 different tests. The most significant differences were in the Morris water maze, which we do a 4-week test, and it gets more and more difficult as the platform moves and gets smaller. The goal is to escape out of this pool using navigational cues around the room. In this case, in the first phase of the hidden platform phase, it turned out to be the high affinity mice that showed impairment. So here, taking a longer time to find the platform is an indication of learning and memory impairments. So over the 6 days of testing, they never quite caught up to their controls. Interestingly, the reverse phase, where the platform indicated by the circle goes to a different part of the pool, we found that it was the poor affinity AHRD that had the most difficult time. So this is an indication of a mouse that has learned the location, and this one is just looking everywhere. So this would indicate impairment, this would indicate learning. So another gene by treatment interaction, but in a different genotype. Then we had Emma Foster, who was awesome and won just a ton of awards this year for her presentations, did the neurochemistry for us, measuring neurotransmitter levels in multiple brain regions. Since the most significant impairments were learning and memory, the data I'm presenting is hippocampus only. And here we found the BAP exposed high affinity knockouts had higher dopamine levels. Conversely, the HRD had lower, but they had higher turnover rates. So these were the two groups that were affected, and they had the greatest difference in neurotransmitter levels. That was also true for serotonin, where the BAP high affinity knockouts had higher turnover compared to their corn oil controls, and you see the opposite pattern in the poor affinity. So to summarize the key findings, the poor affinity AHRD exhibited more anxiety-like behavior. That recapitulated something we found in our PCB research. The BAP exposed females were most susceptible to motor impairments. This is the first time we've seen a sex by treatment interaction with benzoate pyrene, but both knockouts had some level of cognitive impairment, suggesting that CYP1A2 is important in the detoxification pathway, making these mice more susceptible, or potentially the dam, and that's where we'll get the BAP measurements, maybe less is actually being transmitted or transferred over to the pups. And then we saw significant differences in neurotransmitter levels in the same genotypes that were most affected in the behavior studies. Future directions then are with the CYP1A1 knockout mice. We are looking at gene expression currently in multiple tissues, and have some interesting data. A side project Amanda Honecker has started is on cardiotoxicity, and got some in-house funding for that. So that's looking really good. And she's collaborating with some folks over at the University of Cincinnati, working with one of our new chemists, Molly Hopper, on BAP distribution and metabolism. And then I have a couple of honor students looking at interventions. So once you've identified most susceptible, can we do something to alleviate and mitigate the damage? So again, thank you very much to Team Norse, to all of our funders. And if you have any questions, I'd be happy to take them now or at the end.
Awesome. Yeah, I think we should just take a few questions now. I'm curious what ages you test the animals at. I mean, are you eventually going to try and test them when they're younger and then older to see if it's a maintained effect, or?
Chris, we can't hear you.
Yeah.
You're muted, Chris.
There you go.
I was trying to go backwards and it wasn't letting me. So
Oh,
I just
okay.
pulled it up on a different screen.
Sorry, I guess I looked
So
at that. Yeah.
the testing right now is at P120. I know there are people, we do neonatal, and I didn't, with 10 minutes, there wasn't much. So we look at the development of their neonatal reflexes, the writing reflex, the negative geotaxis. We have data on that. But my behavioral mentor was Chip Voorhees over at Cincinnati Children's, and he says it's really better to wait till they're older, because as the brain is developing, you may get artifacts. So I know other people absolutely will test as early as weaning, but we haven't done that. I had with the PCB mice, age them out. So some of these tests are pretty robust, and you can repeat at older to see if they're accelerated aging. The interesting thing there is we found the weights in our PCB mice diverged quite a bit. So weight differences definitely affect their ability to do things like rotorod and swim. So you have that huge confounder, but that's about what we've done so far.
Do you have, I mean, is there any effect on the dams? I mean, do they seem relatively healthy or?
Yeah, one of the most important things we do with our preliminary dosing studies is all of these compounds that can bind the AHR can have some pretty severe effects on heart development, cleft palate, hydronephrosis. So we get the dose down so that the AHR is activated. And one of the reasons we wait until gestational day 10 is we avoid these major problems with organ development.
Yeah.
Hey,
Nice,
Chris,
really interesting.
Thank you.
Chris, do you have any ideas how the differential dopamine level changes in the hippocampus you're seeing relate to the behavioral effects you're seeing?
The interesting thing about dopamine, and we do have some preliminary gene expression data that suggests there's oxidative stress in that area. So excess dopamine, it sounds, oh, we have lots of dopamine, but that can actually, and that's been seen in ADHD and some other patterns, it can lead to increased oxidative stress and direct damage to tissue versus what you saw in the poor affinity where you have less, then that can in fact lead to learning and memory deficits, as well as some people have reported serotonin changes can impair. But they're modest changes in neurotransmitters and relatively modest changes, but significant in learning and memory.
Thank you.
Great, I think maybe we need to move on. Sorry about the short time frame, and have Cindy Corbitt share her slides. Yes. Thanks.
All right. So let me. There we go. Everybody see that?
Okay.
Okay. I like to have my little laser pointer here. All right. So what I'm going to be talking about today is very similar in idea to what Chris just shared. But I'm looking at prenatal cigarette smoke exposure on mouse anxiety-like behavior as part of a larger study that looks at microbiome and metabolic changes and so forth. And I'm sharing this as part of the newly established CAAN lab. We can. I'll explain that in just a second. And basically, I'm in the middle of moving my lab with very little notice right down the hall into a bigger space where I'm combining with other investigators, including Abolins-Abols, that's the AA, and Rachel Neal. So, but just a quick overview of, let me get this out of my way. A quick overview of what's been going on among the CAAN lab participants in the last couple of years. So I've done a project with Deborah Denenfeld of Dancing Well, the Soldier Project, looking at the effects of social dance, kind of like barn dancing, community dance, contra dance. You might be familiar with that. Square dancing, it looks like that. And its benefits for veterans with PTSD. And we did a study that showed increases in wellness for veterans with PTSD who participated in her program. Done a little bit with prenatal valproic acid, which is used as an anti-seizure medication, but also increases risk of things like spina bifida, combined with testosterone postnatally, trying to get at the idea of why autism diagnosis is much more common in males. What is it about being male or having testosterone that increases vulnerability to that? A more recent study in collaboration with my former graduate student, Monica Unseld, who has established the Until Justice Data Partners, and Kira Taylor in Public Health. We're looking at tear gas, and with the protests of the last couple of years and how that is affecting menstrual cyclicity in people who were exposed. And then Mika's Abolins-Abols, who is a new faculty member in our department, is bringing me back to my old love that I did my dissertation on, looking at songbirds and behavioral neuroendocrinology. And this is really all his work. I'm just tangentially associated with this at this point, but looking at nest parasitism, stress, and how it affects parental care. And then my closest collaborator is Rachel Neal. We have pilot funding with the Center for Integrative Environmental Health Sciences here at U of L to look at prenatal vaping exposure, which came out of our work we've been doing that I'm going to talk about today, looking at prenatal cigarette smoke exposure. That's an NIH R15 grant with Rachel, that her main focus has been microbiome and metabolism. I was brought into it because they were finding sex differences in some of the liver gene expression. And that's, with my behavioral endocrinology background, is really where I have focused. And this is a picture of the Teague cigarette smoke exposure apparatus that puffs the Marlboro Red cigarettes and blows the smoke into the chamber. And then we have another chamber with room air. The model here has been established over several years, is exposing mice for 6 hours per day throughout gestation, starting on the first day we see a sperm plug. And we get decreased birth weight and catch-up growth, and then subsequent differences in metabolic pathway, liver gene expression, and also microbiome changes in the dams and in the pups that is long lasting in alpha and beta diversity. All of that is Rachel's stuff. So I'm just reporting that, and I'm going to talk about what my part of this project was, but setting up that as a background for how these animals differ from each other. We also basically are finding something that looks like metabolic syndrome in the cigarette smoke exposed offspring. So why are we still looking at cigarette smoke? Everyone knows cigarette smoke is bad. People shouldn't be smoking when they're pregnant, but they do. So this is looking as recently as 2019, the percent of women who report that they smoked at any time during pregnancy. This is probably underreported because everyone does know that it's bad. But here you might notice that here in Kentucky, we have rates that are in the highest class. So West Virginia still beats us. I don't know that that's something you want to win at, but our region still has very high levels of women who smoke while pregnant. And we know it's bad, right? We already know that prenatal exposure to cigarette smoke will result in early, preterm birth, low birth weight, increased risk of things like SIDS. It's also been found that there are some neurobehavioral effects in infancy, childhood, and even in adolescence, even though the exposure was only prenatal. Some of those effects involve anxiety and internalizing behaviors in the offspring. So that's where this project has come in. So again, the R15 is really designed to look at gut microbiome differences, which we did find, and I've already briefly touched on the metabolic differences. And my role has been to look at the behavioral aspects. Another thing that's been added to this is not only the prenatal exposure to cigarette smoke, but then adding on a high-fat diet in adulthood, in this case, starting at postnatal day 50. Again, it is common that people who were exposed to prenatal cigarette smoke would also have a typical Western diet that includes a lot of fat. And since we're looking at primarily microbiome effects in the gut and metabolic effects, it makes sense to see if that adds additional confounds. So in addition, because they started finding sex differences in the liver early on, and I came on, we're now looking at sex-specific effects of cigarette smoke exposure prenatally, and then adult high-fat diet. So here's the setup that we breed the mice in-house on gestational day 0. When we find the sperm plug, we expose them either to cigarette smoke or the room air, and that is throughout gestation for 6 hours a day. Then at weaning, we separate the offspring into male and female groups, and then at day 50, they are fed a high-fat or a low-fat diet. So we end up with 8 treatment groups with 3 factors: prenatal, cigarette smoke, adult diet, and then sex. So what did I do with this study looking at anxiety-like behavior? So they got switched to the diet around at postnatal day 50. I was looking at anxiety-like behavior assays starting around postnatal day 80. So a month on the diet and extending for another month. We looked at elevated plasmase. So you can't see it very well because the mice are black, but this has open arms and closed arms with high walls. And we put the mouse in the middle. We let them move around for 5 minutes. And we look at kind of brave versus shy. So animals that are brave are going to go out onto the open thing that's raised up off the floor. It's a little scary to look over. And animals that are more anxious or have more anxiety-like behavior will spend most of their time in the closed arms. I'm not going to show you this data, but basically there was very little that we found that had to do with exposure or diet. There are the typical sex differences. We also did light dark box. So this is just showing the light side of the box with a light shining on it. There's a little hole. They can go into the dark side. We put them in the light side. You measure latency to go into the dark, which is where they really want to be. And then you also look at number of entries back into the light side and how much time they spend on each side. Again, there were some sex differences that are typical, but we didn't find any strong exposure or diet induced differences. The marble burying test, we put 16 marbles on the surface of this bedding, let them explore for 5 minutes, and we count how many marbles get moved or buried, which again is kind of an anxiety-like behavior to get rid of this novel stimulus. And then we also did neohypophagia, so eating less in a novel context and a novel food, which in this case was frozen sweet corn that had been thawed. So we did find interesting differences in these two, which is what I'm going to focus on. So again, 16 marbles placed in 2 rows. More marbles buried or moved is indicative of a higher level of anxiety. Didn't find a whole lot with the number moved, but when looking at marbles buried, we found a diet by exposure interaction, where in the sham mice, the ones exposed to room air in both sexes, so here just to orient you, we have our CSE on the left, our sham on the right, and then F stands for female, M stands for male, H for high fat, L for low fat. What we found is that in both sexes, the animals that were on the low fat diet buried more marbles, indicating that the high fat diet was somewhat anxiolytic for them, that reduced their stress. And what we see here is that you don't see that pattern, and you don't see a whole lot of marble burying in the cigarette exposed animals. So in some sense, being exposed to cigarettes made them less anxious, you might say, but eliminated this effect that we saw. Then in the neohypophagia, we recorded the amount eaten in 2 minutes in their home cage, but then again in a novel cage that was new to them. And so again, what we see here is an exposure effect. Again, the cigarette smoke exposure on the left, the sham on the right, and that in this case, the cigarette exposed mice exhibited more neohyperphagia, meaning they eating less in the novel cage than in the home cage, indicating more anxiety-like behavior due to cigarette smoke exposure, and with no interaction with sex or diet. So it's not uncommon that some animals will show anxiety-like behaviors in one test and not another, and that's because different brain regions control those things. And neohyperphagia is linked to the hippocampus, the amygdala, and the hypothalamus. I have had an INBRE next-gen pilot grant that allowed me to look at RNA-seq of the hypothalamus, looking at all three of these groups, and my alarm just went off, so I've talked longer than I thought I did. So I'm going to zoom through this because basically I'm not going to say much. I've got some volcano plots that basically show that we've got several genes that have effects of exposures within sex, effects of diet within sex. Again, it's difficult when you have three factors to look at all three at once with this many genes. But again, the fold change is on the x-axis, I should point out, and this is the p-value here. So, and then we have effects of sex with an exposure, effects of diet with an exposure, effects of sex within diet, effects of exposure within diet. So lots of genes to follow up on. And so that's where we're heading next, is tracking down some of these genes and using things like pathway analysis and Metacore to figure out where all of these interactions are happening, because there are lots of interactions we're seeing of prenatal cigarette exposure, adult diet, and sex. And most of this work was done by Megan Kynard, a previous undergrad. This is also a previous undergrad, Isaiah Burciaga, who's now a graduate student. And again, this is in collaboration now with our newly combined lab that includes Rachel Neal and Mika's Abolins-Abols.
I was kind of curious, I know we need to move on, but I was kind of curious, in the high fat, do you have differences in the weight of these mice, or do you keep the calories the same? It's just high fat versus.
No, yes, there are differences in weight. That's more Rachel's side of things. And
Yeah.
So, and so she tends to present that kind of thing. So yeah, there are differences in weight, there's differences in fat, and there are sex differences in that.
Yeah.
Right.
Wow.
And of course, all the metabolic pathway stuff. And so yeah, the metabolic syndrome stuff, we're seeing sex differences in vulnerability to that as well.
Wow. Lots of, so many things to look at.
Yeah, and we kind of got waylaid by pandemic closures and all of that stuff, and inability to follow up with PCR, which is what we're doing next. And now we just moved our lab like now. So we'll get to it. We'll get to it pretty soon.
Well, cool. Okay, I guess we better move on to Lisa Middleton. Thanks, Cindy.
You're welcome.
Okay, can you hear me, and can you hopefully see the presentation?
Yes.
All good?
Yes, yes, good.
So I am relatively new to the Kentucky INBRE family. I am at EKU, and I am just in the middle of my second year as an assistant professor. Just by way of background, I have a history of doing drug abuse research, and I've studied nicotine, cannabinoids, opioids, and a variety of different models. And I also was a lecturer for about 7 years and have come back into the research world. So I'm essentially kind of getting my lab up and running for the first time. So I wanted to talk to you today a little bit about the focus of the research that we will be doing in my lab. I'm kind of going back to some work that I did in my first postdoc, looking at interactions between the cannabinoid and dopamine systems. I am expanding a little bit to involve here, let me see if I can use my pointer, to involve CBD, which is cannabidiol. And we'll talk about why I'm interested in that a little bit in a minute. So the first thing I want to mention is that THC is really the main psychoactive component of marijuana. And I'm guessing you guys have probably heard of that several times before. But we have learned a lot about THC and how THC affects the brain. And one of the things that my former advisors found out was that when you look at the effects of chronic THC administration in brain, you actually see regional differences in both desensitization and downregulation. And a lot of that is related not only to brain region, but in the time frame through which you see these differences in desensitization and downregulation. So for example, in cerebellum and hippocampus, you can see desensitization within like 3 to 7 days following of THC injection versus a striatum, which is going to be closer to 14 to 21 days before you see desensitization. And the magnitude is different. So the basal ganglia, which can include your striatum and your nucleus accumbens, is going to show lesser desensitization than some of the other brain regions. But one of the things that they've found is that if you look at the caudate putamen, the striatum, you actually see that there's quite a bit of co-localization in those neurons between CB1 and D2 dopamine receptors. And again, THC is a cannabinoid. In brain, we're focusing here on the CB1 receptor. So when we talk about dopamine, we've heard a little bit about it already, but in my area, I focus on how dopamine is related to reinforcement. And so dopamine is considered kind of the main neurotransmitter involved in psychostimulants. That includes nicotine, methamphetamine, cocaine, and what I'm thinking is some with cannabinoids as well. When you look at the main pathways, you can see that there are 2 main pathways for the dopaminergic systems. The first is the nigrostriatal pathway, which starts down here in the substantia nigra, which sends projections up here into the caudate putamen. And then we have the mesolimbic dopamine pathway starting in the ventral tegmental area, and it has a few additional projections caudate putamen to the nucleus accumbens and the prefrontal cortex. And these pathways have been shown in many different studies, not only to be involved in reward, but also in motor and psychiatric disorders. And this includes things like both Parkinson's and schizophrenia. So when we look at those cannabinoids and dopamine together, one of the things that we saw was that not only that has been published, is that not only are cannabinoid and dopamine receptors located on the same neurons, but individually those receptors signal through a G-alpha-IO G protein. And when you activate them together, they actually switch their signaling pathway to a GS pathway. So instead of being inhibitory, they actually stimulate the intracellular signaling pathways to increase cyclic AMP formation. And on top of that, they have been shown to form heterodimers. So these guys clearly are somehow interacting with one another. Now, if you look at an anandamide transport inhibitor, and anandamide is going to be an endogenous cannabinoid, if you inhibit that anandamide transporter, it actually reduces some of those behaviors that you would see from D2 receptor activation. So again, it seems like there's this overlap between how the cannabinoid and dopamine systems work together. So again, if we look at endocannabinoid release from a different variety of neurons, it requires activation of D2 receptors. So again, we're just seeing over and over again that somehow this relationship between those 2 systems has to be important. So previously in my postdoc, I did work in CB1 receptor expressing CHO cells, a cell line that I had worked on and produced. And what I decided to do in my lab to really examine the connection between CB1 and D2 receptors was identify the SHSY5Y cells, these right here, which are a neuroblastoma cell line that natively express both CB1 and D2 receptors. So I was hoping that this could be a good model to look at those interactions between those systems, since they're already present and clearly will have something that they are working on together. Now, the goal here, since I'm in my first year and a half, was to get these cells, which was fun during COVID, and get these cells growing. They are very different than other cell lines I've worked with. And I was very fortunate to have a graduate student my first year who literally got my lab up and running and helped me get these cells growing and some preliminary experiments going. And my goal was to examine the interaction between those cannabinoid and dopamine systems following both acute and chronic cannabinoid administration in combination with a dopamine agonist. So trying to see if activation of those receptors change signaling in these cell lines. And I decided to take it one step further. CBD is a drug that you probably have all seen in your local gas stations. You see it in your pharmacies. You can take it as an oil. You can take it as a salve. You can take it pretty much as anything you want. But CBD, and I'll tell you in a second, also has a role in this cannabinoid and dopamine signaling. And it's often taken by people who are either using THC or using dopamine agonists. So the interesting thing about cannabidiol is that it does not have any psychoactive properties. So it does not show any of the same effects as THC, including behavioral, motor effects. And it is the second most abundant cannabinoid in the cannabis plant. So there's a lot of it, but a lot isn't known about what it does. We see reports of people using it as an anti-inflammatory, for pain, as an anxiolytic, and even as an anti-tumor medication. But really, there isn't a lot known about how it's working. The cool thing is it has very few side effects, and there's no substance dependence. There's been no reports at all of dependence. However, with CBD, it is highly variable. So there is no real regulation across any oil that you might take at any different pharmacy, gas station, etc. Everything is going to be changing and evolving because now it's an over $1 billion industry. So people are getting in on the ground floor trying to get in on CBD. There is currently only one FDA approved product called Epidiolex, which is considered a Schedule 5 drug. And that is the only FDA approved CBD containing product. Right now, it is not regulated through the FDA because it is considered, what is the word I'm looking for? It is considered a supplement. So they can say it may have these effects, but we can't guarantee it. So there's really, really no control over the CBD production. The one main thing is, though, your CBD that you can get at the pharmacy or the gas station cannot contain more than 0.3% THC. So they do have to try and make it relatively pure. As I mentioned, its mechanism of action has not been fully elucidated, but we do know that it actually has a really low affinity for the CB1 receptor. And in fact, it could be acting more as like a CB1 receptor antagonist, which is interesting because it's present in the native marijuana plant. So you're smoking the THC and getting the CBD, and it actually may be conflicting against what the THC is doing. But we've also seen that it is a partial agonist at D2 receptors. It works at serotonin receptors, opioid receptors. So it actually is kind of messy, and we're learning a lot about what the mechanisms of action are. So with regard to our treatment conditions for our initial setup, as I was preparing these experiments, I was like, this is why people don't do a lot of polydrug use experiments, because you have a lot of treatment conditions to make sure you're properly controlled. So we are looking at CBD alone, the D2 agonist quinprol alone, and the CB1 agonist WIN55212-2 alone. And then we're combining those drugs CBD with the D2 agonist, WIN with the D2 agonist, and all the varieties of combinations. I will tell you that we picked WIN 55212-2 because it is a full agonist at CB1 receptors, and our department does not currently have DEA approval and set up so that we can house THC. So that is something I will hopefully be working on over the next couple of years to get that licensure so we can actually test THC as well in these experiments. Now, I also will tell you that as I had my graduate student with me and we were getting her thesis finished, I actually chose high doses of CBD, quinprol, and WIN to test first. We need to go back and do some dose response studies, but I wanted to start with the high doses, doses that have been shown to produce effects in other studies and in the lab that I worked in previously. What I'm going to show you is very, very preliminary, so I'm going to go over it relatively quickly. But in this slide, I just wanted to show you that we have indeed been able to perform Western blot analyses on these SHSY5Y cells. And at the top here, you will see this band across each of these lanes represents the CB1 receptor. G protein coupled receptor antibodies can be a little dicey to work with. We have found an antibody that appears to work for now to identify the CB1 receptors. And I just think it's important to recognize that indeed these cells do have those receptors. We also see here that these cells represent or express the D2 receptors. So here is our D2 receptor band, and this is a native expression. We did not alter any upregulation or signaling pathways to get these to express the D2 receptor. Obviously, we have some things and kinks to work out. We have our beta tubulin down here, our concentrations we need to work out. We need to work on our protein loading, but my grad student was able to do all of this work in a very short amount of time. So we have a cell line, a mechanism to actually see both CB1 receptor and D2 receptor activation in this single cell line. We also are going to be using confocal microscopy to determine whether these drugs alter receptor localization. Sometimes you can see downregulation, but more than anything, we've seen in previous studies that you see alterations in localization. And so this is, again, a very preliminary study where we had, we stained these cells for both DAPI and the D2 receptor. And in red, you can see that the D2 receptors are present. In blue, you can see that the DAPI is present. We have cells with nuclei, and we are working on optimizing conditions to really, number one, plate the cells properly, get them to stick, and stain them properly. The CB1 receptor is a little bit trickier to work with regard to checking or activating the receptor, staining the receptor. So we're still working through getting that done. And also we're looking at cyclic AMP activation. Remember, if you stimulate with WIN alone, it should decrease cyclic AMP. When you stimulate with quinprol alone, it should decrease cyclic AMP, and both of them together should increase cyclic AMP. So we have started the process of measuring cyclic AMP levels following both acute and chronic drug administration. I'm just showing you that we have begun here collecting data. We will need to repeat these data because we learned a lot about this assay. It is new to me and to my lab. And we did have tons of cyclic AMP and need to modify our protein concentrations and cell concentrations a little bit as well. And so ideally what we'll be able to do is complete those dose response curves to look at both high and low concentrations of our different drugs of abuse or different drugs. We also want to include protease inhibitors because we did notice that some of our lanes were a little dirty, particularly with the CB1 receptor. We're hoping to clean that up by using a protease inhibitor, finalize some of our co-localization with CB1 and D2 receptors. And one of the things that I would long-term like to look at is specific G proteins in these SHSY5Y cells to determine if those play a role in the change in signaling. I'm just going to quickly mention this little guy right down here. as part of my Kentucky INBRE Idea Award application and awards that are available through NIH. I am also hoping to expand into the neuroinflammation area, an area that's of high priority for NIDA, to see if we can see differences with these pathways in neuroinflammation. I also have a contact at a drug company in Lexington that produces CBD. I know I'm done with my time, I'm hurrying. They produce CBD, and I have been offered CBD products for free at various levels of production. So various levels of purity as well. So I am interested in pursuing that further to see how these different CBD products really can impact signaling. So all of this work was done in conjunction with Hope Dell, who was my master's student. She was an absolute workhorse and literally got my lab up and running during COVID. It was amazing. Jenna Stewart was an undergrad in my lab. I could not have done any of this without Lindsay Cormier and Brad Kramer in our department, who let me use all of their equipment because, again, COVID. And then again, this was funded through Kentucky INBRE. So I will stop rambling there.
Great. That's very ambitious. You got a lot of things going on.
It's a little busy.
Can you tell us a little bit more about that, the cell line? Is that from striatal cell? I mean, how is?
So it's the neuroblastoma. I think it's from bone marrow cells. So again, it was chosen based on what it already had available. It prevented me from I have previously created a CB1D2 receptor co-expressing CHO cell line.
Yeah.
But this lets me kind of jump in and immediately have a cell line available. Grow these cells. I did not do any additional treatments. I know that if you add retinoic acid, you can help them differentiate more into a dopaminergic cell line. And that's another thing that we've talked about adding.
You think you'll go back to your CHO line, or is that an option?
I love the CHOs. They are so easy to work with. They grow so fast and they're so hardy. So literally any undergrad could pick up and work with them. So it is not out of the realm of possibility that we will try and go back to those. I would like eventually to take this, you could take it for sure into a mouse model and look at different brain regions and do Westerns and confocal that way as well. So there's a lot to think about.
Yeah, lots of things that you can, lots of directions to go. Well, thank you. Any other questions before we have our last speaker?
Oh, sorry.
Xiuhua? I'm sorry. You've told us
you're
before.
fine. Yeah, you're completely fine. Yeah. I actually so surprised you still remember my name. Like, oh, I think I need to switch. Can you see the presenter?
Yeah, I remember you on your last slide before when you spoke, you gave us the pronunciation.
Yes. So I have my pronunciation here, and I'm sorry with my face, and I have to close my office door and to work because of COVID and it's hot in the so my name is Xiuhua Ding, and I'm from the Department of Public Health from Western Kentucky University. And just a little bit brief background. My major in epidemiology and biostatistics. So my research background is focusing on observational studies related to Alzheimer's disease or the dementia. So here's some background of the Alzheimer's disease and the dementia. I think all you know. So dementia is more a symptom that affect cognition affect emotion, behavior, and also as the ability of daily living. So the common causes for dementia is the Alzheimer disease. So it's account for 60 to 80% of dementia cases. And Alzheimer disease is a type of progressive brain disease. So it usually happen to a long time. So the early symptom may include difficulty to remember names or events or documents or some kind of as early symptoms. Then it goes then it's going to get worse as disrupted communication, languages, also prejudgment or disorientation. And then eventually they're going to have difficulty speaking or difficulty to living, difficulty to daily life, such as dressing themselves or eating. And then eventually probably they are bad bound and need intensive care. So the prevalence for the Alzheimer disease is around 6.2 million in the US. That's more than 1 in 9 people for age 65 and older, especially for the 75 years older. That's 75% of cases from cases from 75 or older population. So there, with worldwide population aging, the projected prevalence of the Alzheimer disease dementia will increase, is predict going to be double by 2050, and it's going to be around 30.8 million around by 2060. So what's the impact of the Alzheimer disease dementia is, first of all, is 1 in 3 senior dies from Alzheimer disease, and it's already officially listed as the 6th leading cause of death in the United States. From the figure in the right, you can see other causes of death decreasing between 2000 and 2019, but only the Alzheimer disease death increase between 2000 and 2019. So not just the cause for death, there are other causes from the family giver, the caregivers, and family and the friends, and also cost for health care and the long-term care or hospice. So their estimated unpaid cost from friends or caregiver is about 257 billion last year, and its total cost, estimated cost from the health care, long-term care, and hospice is 355 billion dollars. So those numbers are going to increase over time because of the prevalence increase. And those costs from the caregivers, including bathing, like bathing, shopping, and also emotional and social support. So that's the challenge from the dementia. When we're looking at the Alzheimer disease dementia, we have this genetic factor, APOE. It has been, there are many studies showing is a has a strong impact on the risk of late onset Alzheimer disease. And everyone will have one of 6 possible genotypes or APOE pairs. You either be E2, 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 E3, E2, E4, E3, E3, or E3, E4, and for or E4, E4. So for the people who have E4 allele, they have increased the risk of developing Alzheimer's disease. And here are some numbers. For compare people with E3 form, and the people have one copy, which means the people with E3, E4, or E2, E4, those people will have 3 times the risk of developing AD. And with compared to people with double E3 and 2 copy, people with 2 copy of E4, that will be people with this genotype, they're going to have 8 to 12 times of the risk of developing AD. And there's some literature found the APOE APOE cause dementia probably because it's associated amyloid beta degradation. So since APOE is unmodified, risk factor is genetic factor is unmodified. And AD is not curable or even not treatable. So researchers have been looking for modify risk factors to kind of prevent or slow the progress of the AD. So one of the promising modifiable risk factor so far is the sleep apnea. So the symptom of the sleep apnea, including snoring, breathing pauses during sleep, headache, and daytime sleepiness. So it's more common in older population. And there are some signature EEG abnormalities showing in people with mild impairment, mild cognition impairment shows reduced slow wave sleep and fewer spinal needles for the people with sleep apnea. So those are some kind of basic general background. So there's some research we did. We have some previous studies showed their heterogeneous effect of APOE on the risk of dementia. The risk of dementia depends on the status of sleep apnea and APOE. So people with we found in US males, people people have the APOE with E4, with people with the APOE E4, there is no increased risk between sleep apnea and dementia. But for the people without APOE E4, there is increased risk if subject has the sleep apnea. So for this interaction, so those interaction study were not is understudied. So it's not. There are some limitations, and the mechanism explain the interaction among sleep apnea, APOE, and the risk of dementia are not clear and inclusive. So I use this figure from this article and to kind of discuss the possible mechanism of the interaction between APOE and sleep apnea, maybe because they are the sleep apnea, maybe people with the sleep apnea, maybe they have intermittent hypoxia and also oxidative stress, or also there's some inflammation. So those are the factors maybe are associated with amyloid beta production or release, or even aggregation. So, and also APOE is associated with amyloid beta degradation. degradation. So so maybe they share some pathway. So it's worthwhile to investigate. So so we designed this study to have three specific aims. One is replicate our previous study to verify with improved measurement, improved sample size, and to verify if there is a if there is a interaction on the revised with revised data. And also we want to examine at the baseline whether APOE is associated with sleep apnea for the people without dementia or they are cognitive normal subject, to see whether sleep apnea APOE is going to increase the risk of the dementia, or APOE is associated with sleep apnea alone. The third one, we want to find out the mechanism whether their amyloid beta is more associated with sleep apnea and APOE. And also we want to look at the trajectory of the amyloid beta changes over time for the cognitive normal subject before they develop to the dementia. So our research plan is we use secondary data analysis, which the data was from the National Alzheimer Coordinator Center from Washington, Seattle. So the center was founded by National Institute of Aging in 1999. They actively collecting or recording data from the NIA-funded Alzheimer's Disease Center nationwide. And so they here all the national centers in the dark blue, that's a NIA designated centers, and they actively collecting data since 1999, and they are going to follow those patients or volunteers annually to get a longitudinal or follow-up data. So for this specifically study, we extract the data from 2015 until 2020. That's we extract all the data for the people without the dementia, or at 2015 or after 2015. When they enter this database, they are cognitive normal, and we are going to follow those up, follow those subject up to see the time they develop the dementia. So we only have some data on our specific aim, the second specific aim to look at the APOE, whether it's associated sleep apnea, even they are cognitive normal. So here the subject information, there are 3,846 qualified and then or eligible, and their age about 868, and they are about 34 males, and their education, they are kind of highly educated population. They're 14% of them are APOE4 carriers, and in normal population, or they are about 25%. So here we run the statistical modeling, and we're looking at the logistical regression to compare, to look at adjusted after adjusted confounding or confounding variable, and to see whether APOE is associated with sleep apnea. We got our final models. APOE is not associated with sleep apnea at the baseline for the people without dementia, which is more, which is similar as the conclusion from other studies. and we submit this abstract to the Alzheimer's dementia or Alzheimer's AAIC Alzheimer's Association International Conference in 2020. That was funded by the KBRIN for 1 year new pilot study, and we kind of update our basic aims and for resubmission and their development, including sharpening the specific aims. Also, we kind of applying some novel method, use some novel statistical method or epidemiology method, use data harmonization, including add another, pull the data from the Alzheimer's disease coordinate center data and with the ADNI data, and found more collaborators for this project. Unfortunately, I did not get this, but I think it's really helpful for the I look at the review, really helpful. I think for the future direction, I'm planning to apply for R15 in the October, and I think the direction we are going to recruit the subject maybe through the sleep clinic here in Bowling Green, also probably in some from the Vanderbilt clinic, from the collaborator in Vanderbilt, and we focus on more applied solution or therapeutic solution for the disrupted sleep, maybe to see if that influence the amyloid beta production or clearance, to kind of slow dementia, or also looking at the diagnostic solution for the dementia. So that's all my.
Nice, thank you.
Thank you.
Any questions? I know we're a little over time.
The underlying factor of all this just be hypertension, since APOE high amounts causes hypertension. Hypertension causes sleep apnea and therefore has a very big effect on Alzheimer's. And so have you looked at whether it's just actually the hypertension is the root cause?
Yes, hypertension. So if you look at the model, my model actually, my model included hypertension and diabetes. So those are the factors, the cardiovascular disease factors or increase the cardiovascular disease. Those factors actually are associated with the risk of dementia as well. Yes, yes. But since I did not talk more about that because I want to just kind of focus on sleep apnea and APOE here. But it's definitely hypertension is one of the risk factors as well. And also I did not see the education. It is the education is one modifiable like factors, like if you read more or you work more when you're older, you probably have a later onset or lower risk to develop dementia as well.
Okay, well, I think we better, any other questions? I think we might need to wrap it up because we're a little over time. I appreciate everybody hanging with us. Thanks, everyone.
Thank you.
And we'll see you next time. Here, let me.